Okay, so hello and good afternoon to everyone. Today we have in our Young Investigator seminar series, we have a special guest who is Francisco Esteves. He spent some time in Edinburgh with Lenny Nelson, who is now trying to connect. I will send him immediately the link. And he will tell us a little bit what was the purpose of the short-term scientific mission that he performed in Edinburgh. I think he had some good techniques with the HepaRG cells that Lenny uses quite frequently. And Francisco, maybe you can start while I give access to Lenny. Francisco, are you there?
Yes. Okay, I will start to share.
I see you now. Perfect.
Hello?
Okay, can you see my screen now?
Not yet.
I see you frozen. It's opening now. Perfect now.
Perfect.
Okay.
Hi, Maribel.
Hello.
Hi, good afternoon. So, with no more delays, I think I can start my presentation. If you allow me, Javier.
Yes, for sure.
Okay.
I'm just trying to connect Lenny so he can see you as well.
Okay. So, thanks for the introduction. Thanks also for the invitation and the opportunity to share my research and also my work developed during my short-term scientific mission at Edinburgh. My presentation is titled STSM Training in the Use of HepaRGs and Monitoring Pore Expression from Lisbon to Eddie and back. The presentation is divided in three main sections. An introduction on the importance of cytochrome P450 oxyreductase or CPR on chain-abiotic metabolism. Then I will talk a little bit about our research, our forming and ongoing studies on the role of interaction of CPR with its redox partners. And finally, I will focus on our main theme of the presentation, which is the STSM, all the scientific gains, also the strengthening of collaborations and our future perspectives. The human cytochrome P450s or CYP constitute a superfamily of monoxidase enzymes with 57 isoforms with high degree of genetic polymorphism. As you can observe in this table on your right, you can see the distribution of the CYP isoforms by their main substrate type. And microsomal CYPs are depicted in black, while mitochondrial CYPs are depicted in gray. CYPs constitute an important class of drug metabolizing enzymes for their central role in chain-abiotic metabolism, but they are also important in the other pathways, for example, endobiotic metabolism, such as steroidogenesis. Importantly, the activity of all CYPs is strictly dependent on the redox partner. While macrosomal CYPs are strictly dependent of the transfer from NADPH through CPR, or cytochrome P450 oxyreductase, mitochondrial CYPs utilize adrenodoxin reductase and adrenodoxin to get their electrons. So this is an important difference between microsomal and mitochondrial CYPs. So why study CPR? Well, CPR is a central enzyme in several CYP-mediated reactions, such as chain-abiotic metabolism, but also in steroid synthesis, retinoic acid metabolism, and sterol synthesis. CPR is also important in other known CYP-mediated reactions, such as cholesterol synthesis, cytochrome B5 reduction. CPR can directly bioactivate some chemicals, and of great importance also, CPR is the obligatory donor of electrons to heme oxygenase essential in degradation and also on carbon monoxide and iron homeostasis. So CPR is a kind of central hub control in several archaic physiological process in endoplasmic reticulum. CPR also demonstrates a very interesting protein dynamics. Here in this figure, you can see the model of the open-closed dynamics of CPR in its electrotransfer, and in its closed conformation, we can see that the FMN domain here in blue and the FAD domain interact, and this produce an interflavin electrotransfer. While in the open conformation, the FMN domain here in blue, it's completely accessible to external receptors, and the FMN domain CYP interaction occurs, and there is an interprotein electrotransfer. Studies from our group and others showed that there is an ensemble of open conformations of CPR for interaction with structural different and diverse redox partners. These open conformers are somehow sampled by diverse redox partners, which is determinant for an effective electrotransfer. And how this CPR-CYP interaction occurs. We know that basic residues on the proximal side of CYP isoforms form a positive electrostatic ring that steers the docking to a negatively charged patch on the FMN domain of CPR near the hem of CYP. However, the exact mechanisms of protein-protein interactions within the CYP enzyme complex are still not fully understood. While in mitochondrial CYPs, there is a signature, as you can see in this alignment of residues, there is a signature of conserved K amino acid on the proximal surface for interaction with adrenodoxin, which is the electron supplier of mitochondrial CYPs. In microsomal CYPs, there is no specific protein structural feature or amino acid signature in CYP proximal site. Well, probably there is a degenerated signature of CYP surface amino acids on its CPR binding interface. Several studies suggested that the CPR mutations may cause a CYP isoform specific effect. So the question is how CPR interacts with all these structural different microsomal CYPs for an effective electrotransfer. We hypothesize that CPR interacts with microsomal CYPs in an isoform dependent manner. We know that differential affinity parameters of microsomal CYPs for CPR may be a key element in the assembling of CPR with a specific CYP isoform. So if we are able to modify the FMN domain surface residues of CPR for increased activity of a particular CYP isoform in a kind of specialization, we'll be able to identify the critical residues that are determinant for CPR's affinity for CYPs. For that, we created CPR mutant libraries targeting the FMN domain in collaboration with INSA Toulouse in France. We use high throughput assays after validation to characterize the critical residues and looking for this CYP binding interface of CPR. Our first objective was to decipher the role of the CYP binding interface of CPR in the FMN domain in the CPR-CYP interaction and how it determines the fate of electron transfer to multiple structurally different redox partners. The steady state kinetic parameters confirm the gain in CYP activity for 7 CPR mutants from more than 2,000 candidate CPR mutants that were in combination with specific CYPs. These 7 CPR mutants are described in this table here on the right. We were able to identify 3 mutants in combination with CYP1A2 in green. Other 3 mutants in combination with CYP2A6 in blue, and finally 1 mutant in combination with CYP3A4. These CYPs were chosen because they are representative of the main CYPs families in drug metabolism. In the graph on your left, you can observe that the relative activity of these 7 CPR mutants were statistically higher when compared with the CPR wild type. So CPR FMN domain mutations may induce higher affinity of CPR to specific CYPs, showing the importance of the study residues in this degenerated interaction. The structural analysis of the FMN domain mutants of CPR that you can observe here in these two figures. On your left, the protein sequence of the human CPR FMN domain, and on your right, the crystal structure of the FMN domain. We observe that the mutated residues that show a higher affinity to a specific CYP that are depicted in the figures in blue, they colocalize with acidic stretches that were previously suggested to be involved in CPR-CYP interactions that are depicted here in magenta. So this colocalization confirms the importance of these stretches. Also, they colocalize with tyrosine residues that are involved in the FMN binding, here depicted in gold. And these mutated residues were also in the vicinity of naturally occurring CPR variants that previously were suggested to have a CYP isoform dependent effect that are depicted in green in the figures. So all this data led us to hypothesize that this CYP-CPR interaction is dependent on a CYP isoform specific combination of several binding motifs on the FMN domain of CPR. We know that microsomal CYPs turnovers can be modulated by the efficiency of the complex formation. So considering that, if we combine the FMN mutants that previously were shown to increase the activity of a particular CYP, if we combine these mutants with other different structural CYPs, we'll be able to confirm the CYP isoform dependent nature of the CPR-CYP interaction. So considering this, we selected specifically 4 CPR mutants that previously showed increased activity of a particular CYP. We recombined these mutants with other CYP isoforms, and we assessed these CPR mutant CYP couples activities, looking for this degenerated interaction. So our second objective was to confirm the generated FMN domain mutants of CPR are somehow specialized for a specific CYP isoform, while detrimental or without impact for interaction of CPR with other CYP isoforms. Here you can observe the plots of relative activity of the different CPR mutant CYP combinations. We have the 4 mutants, CPR mutants, in combination with CYP1A2, CYP2A6, and CYP3A4. And the results confirmed the increased activity of specific CPR mutant CYP couples that were already previously showed to have increased turnover rates. These 2 mutants with CYP1A2, this mutant with CYP2A6, and this mutant with CYP3A4. But the most important results from these experiments was that the new CPR-CYP couples, except for one, this mutant at position 175 in combination with CYP3A4, but all the others demonstrated equal or significantly lower activities when compared with the wild type. So, in conclusion, the single mutations in the CPR FMN domain may cause an increased electron transfer efficiency for one specific CYP isoform while maintaining or even decreasing this activity for other CYP isoforms. The molecular dynamics modeling of CPR domain mutants showed that the 4 mutations do not perturb the overall structure or folding of the FMN domain, but nevertheless, we were able to detect subtle deviations that were changes that propagate to the electrostatic potential with specific and localized effects. We observed a decrease in charge density in the region near the mutant at position 117 when compared with the wild type, and also an increase in the negative potential in the region of the mutant at position 151, of course, when compared with the wild type. Also, a decrease in this negative potential was found around the FMN moiety and was caused by the mutation at position 144. And finally, the mutant at position 175 induced a subtle deviation of the residues surrounding the FMN moiety. From these observations, what is important is that mutants that were isolated with different CYP isoforms, they cause different or even opposite effects at the same area of the FMN domain. This is indicative that CPR establishes effective complexes with its structural diverse partners through a combination of specific structural features of the FMN domain in a one-fold type of interaction. Also, genetic variance in the identifying binding motifs of the FMN domain of CPR may have physiological implications in biotransformation. And this data was my road to Edinburgh. Well, with these molecular results, we intend to perform the verification of these molecular findings on CPR variants in CYP-dependent metabolism, but in a human hepatic cell model. And this was my ticket to Edinburgh. I went to Edinburgh to Dr. Leonard Nelson lab at the Institute of Bioengineering at the University of Edinburgh. In March 2020, just in the early beginnings of the COVID-19 pandemics. And I will start the introduction of the STSM by talking a little bit of CYP-mediated metabolism and DILI. All we know that drug metabolism is highly dependent on intrinsic and exogenous factors that are determinant in CYP relative expression and activity. Especially these expression and activity of CYPs are highly dependent on the genetic background of the subjects. Also, genetic polymorphisms in CYPs were associated with DILI and identified in several candidate gene studies already. Here you can observe the classic pathway of acetaminophen or paracetamol, and we know that when there's a saturation of glucuronidation or sulfation due to an overdose or even a chronic liver disease, a percentage of acetaminophen is metabolized and bioactivated through CYPs activity. Well, we have the synthesis of NAPK, which is highly reactive metabolite with high hepatotoxicity. So if you have in mind that this pathway here is highly dependent on CYP activity, especially CYP2E1 and also 1A2, and you think that all these CYPs are highly dependent on the activity of CPR because all xenobiotic metabolism CYPs are strictly dependent on their activity, are strictly dependent of CPR activity and electron donation, we have to make the question, In what extent does the genetic variability of the POR gene encoding CPR leads to an altered CYP-mediated drug metabolism? And to answer this question, we intend to use the HepaRG in vitro hepatic model, which is considered a surrogate for human primary hepatocyte, and a robust organotypic co-culture model of human hepatocytes and cholangiocytes, with evidence of hepatic polarity, maintenance of in vivo-like liver-specific functions, and substantial drug metabolism levels, as you can observe on your left in this table. The expression and activity levels of functional phase 1, here several CYPs, and phase 2 DMEs, these activity levels are comparable with the ones of the primary human hepatocytes. Also on your right, you can observe the comparison of intrinsic clearance of HepaRGs and hepatocytes for tolbutamide, which correlates very nicely. So HepaRG is a nice model, and we so have a general hypothesis for this STSM that was that CPR variants play a central role in CYP-mediated drug metabolism, and we may assess these CPR variants and their effects in CYP-dependent drug metabolism at an hepatocellular level using the HepaRG cell system. So the objectives of this mission were the gain of experience in handling the HepaRG cells, but also development and optimization of specific assays to monitor CPR activity. And this STSM was also used as successfully used as a primer for strengthening collaborations with our lab in Lisbon and lab of Dr. Leonard Nelson in Edinburgh. As achievements, we were able to differentiate cells from undifferentiated HepaRG progenital cells during my stay. Also, we developed some phenotypic and activity tests. We were able also to perform the isolation of subcellular fractions and study the expression of some DMEs. And this allowed knowledge exchange through training in HepaRG cell biology and cultural techniques applicable for DILA studies from the lab of Dr. Leonard Nelson in Edinburgh to our lab in Nova Medical School in Lisbon. Also, we were able to assess CPR activity using the cytochrome C reduction assay that was redesigned and optimized to a high throughput 9612 microplate format. This cytochrome C reduction assay is based on the fact that cytochrome C is a non-physiological electron acceptor of CPR. And when in its reduced state, cytochrome C has a max absorbance peak at 550 nanometers. So this reduction can be measured in time by reading absorbance at 550 nanometers. This assay allows us to calculate and determine the CPR content in samples of microsomes isolated from differentiated HepaRG cells. This allowed the knowledge exchange through development and optimization of enzyme activity assays applicable to DILI from our lab in Lisbon to the lab at Edinburgh. Future perspectives. We are now developing some metabolic studies with HepaRG CPR variants in collaboration with Dr. Leonard Nelson. We intend to use CRISPR-Cas9 to edit the HepaRG genome to obtain CPR mutants and natural variants, and also a CPR knockout cell line that will be used as a control. We are developing, and we start already developing these assays during my stay at Edinburgh using standard fluorescence probe substrate kinetics. We intend to study the differential activity outcome of these CPR mutants using substrate compounds with well-defined metabolic profiles, for example, caffeine or clozapine, that are known to have multiple CYP-dependent metabolites. And finally, we intend to study the impact of these CPR mutants in hepatotoxicity by using substrate compounds known to be somehow implicated in DILI, such as, for example, paracetamol. All these experiments will be performed ideally using HepaRG 3D models when, of course, possible. The expected outcomes. Well, we intend to assess the HepaRG CPR variants capacity in sustaining CYP activity, and also the effects of these variants in altered toxic effects in drug metabolism. And also we intend to understand the basic mechanisms that underlie chemical stress mediated adverse drug reactions in humans, in particular those related with DILI. We already developed and designed a collaborative project titled The Rule of CPR Protein Dynamics in the Variability of Drug Metabolism and Adverse Drug Reactions. This project was also designed to be in collaboration beside our two labs, in collaboration with INSA Toulouse in France. And with this, we'll be able to study the translation of molecular findings in the characterization of the rule of variability in CPR protein dynamics in xenobiotic metabolism on the hepatocellular level. This project is waiting for funding. Unfortunately, last year, due to the constraints of COVID-19 pandemics, the project was not able to get in fund, but due to a very positive evaluation, we'll try this year again and expect to have funding to work on this subject. Also, this STSM has led to a strong collaboration between our lab at Nova Medical School and the lab at the University of Edinburgh, and of course other groups within the Pro-EuroDILI network working towards major grant applications as well as publication in this area. Who knows, as a spin-off of this cost, a Pro-EuroDILI network, who knows? As final remarks, well, we know that CPR is important in xenobiotic metabolism, but we also have to consider that CPR is central in steroidogenesis, in sterol synthesis, in fatty acid metabolism, and also of great importance in heme degradation. And all CPR variants should be studied, and the implications of these variants should be assessed in these pathways also. Finally, I would like to thank all my colleagues at Nova Medical School, Toxomics, especially to my PI, the PI of Xenobiotic Metabolism Lab, Michel Kranendonk, also a member of this cost. Also, I'd like to thank colleagues of molecular and metabolic engineering team at INSA Toulouse, France, especially to Gilles Drouin and Philippe Ourbin. A very special thanks to Leonard Nelson and Elisa Martin Perez for their kindness in making me feel at home during my stay at Edinburgh. Also, special thanks to the Pro-DILI network coordination that allowed me to attend to several initiatives that were very important for me. For example, last month at the Faculty of Pharmacy in Lisbon, the training school on toxicology, that was very interesting. And also special thanks to the STSM coordinator, Javier Cobero. Finally, I hope you're still awake, and many, many thanks for your attention.
Thank you, Francisco, for this super interesting talk. Actually, it's a topic that I didn't know so much in detail, and the polymorphisms look really fascinating. So, how effective is the CRISPR-Cas deletion in HepaRGs? Is it like a real knockout, or is a?
Well, till now there are two approaches, possible approaches, and one approach is from a group from Germany. They were able to get about 90% of knockdown of the CPR activity. So also there is another process that may be used using paracetamol, because with paracetamol selection of CPR, for example, for the CPR knockout, if you don't have CPR, you don't have CYP activity. So the still working cells, clones with active CPR will be not able to proceed because of the toxicity of NAPK formation and the selection of the cells that are completely knocked out. But yeah, it's a challenge, of course.
It is, but I think it will. I mean, 90% is already like a knockout, so it's quite good.
Yeah,
it's quite good. Do you any of you have any other things to Lenin? Do you have any comments on Francisco?
No, it was perfection.
Hi, Lenny.
Hi, Francisco. How are you?
Very fine. Very fine. How are you?
Oh, living the dream. That's me. You know, yeah, great talk. Fantastic. I learned a lot there again, as I did, you know, from your stay. And it was, you know, the work you're doing is really first class, actually. And so I was just thinking about, you know, your earlier slides on the, you know, characterizing the biophysical aspects of CPR, the POR gene, the human POR gene, and all this beautiful, intricate, elegant, delicate work. I mean, what's the translational aspect of this? Apart from, you know, basic science, we're getting the biophysics, you know, biophysical characteristics of the CPR. What's the translational angle? Is it, you know, can you design drugs from this by artificial intelligence or something?
So you are talking about the variants, the CPR variants?
Yeah. Well, say the variant. Can you design?
Our first objective was to trying to determine the motives, the binding, the structural features in the FMN surface that were determinant for the interaction with different CYP. That we know that CYPs is a family of highly variant, structural variant enzymes, and how CPR is able to interact with all these different enzymes and is capable to give the electrons. This was our first approach. Of course, with the following of the work, we also observed that some of these mutants are near the variants, natural occurring variants in the population, and that may be involved in differences in the activity of CPR. Okay, so it may have impact in the biotransformation and of course in adverse drug reactions and of course ultimately in DILI. I don't know if I answer your question.
Well, I was thinking more in drug discovery or something. Can you design small molecules against these specific binding regions and pockets for addressing, you know, some of the diseases associated with CPR deficiency, like anti-Bixler syndrome and all that?
Probably you can design some drugs or use new techniques of gene editing, trying to fix some problems that CPR may have in some subjects. Yes, why not?
Can I interrupt on that? And
yeah,
just
of course, Michel, please.
Add on to this what Francisco said. I think it's important to understand what he said, that knowing that these polymorphisms occur in the human population and with a frequency that is not that low, meaning that there might be people that with certain CYPs might have different activities with if they have this CPR variant. So you might think about a different metabolic outcome for particular drugs. Responding to your question, maybe designing molecules that might interact, and I think that might be very, very difficult. The stretches on the CPR-FMN domain, they are very specific and they are very close. So it might be possible, but I think you will have, if you want to direct it for the interaction of the CPR mutant with a specific CYP, you might have a spillover affecting also the interaction of other CYPs, because it is a combination of the binding motifs, and in one CYP, one particular binding motif might be a little bit more important, but for another one it is it's still interacting with another CYP. So if you will have a pleiotropic effect probably. So I think in that particular aspect, I think it will be very difficult to direct or design a molecule that will prevent a specific part of the binding motifs within interacting with specific CYP.
Yeah.
But also thinking physiologically in the CPR activity, you have to take in mind also the dependence of hemoxygenase on CPR activity. And we know that hemoxygenase is involved in iron and carbon monoxide homeostasis, and that the levels of iron are highly implicated also in several processes in cell cycle like ferroptosis and others. So it is also interesting to study these CPR variants in looking also to other pathways other than the drug metabolism by CYPs.
Yeah, I mean, it's involved in steroid metabolism and, and quite a few. It's almost sort of pleiotropic type functions.
Yeah.
Anyway, yeah, so thank you.
Thank you, Lenny.
Francisco.
Hi, Joanna.
Hey, maybe I can also, I mean, it's actually, I'm a total ignorant on CPR. I was not aware of all of this information, so it's great amount of work. I just wonder, this is ignorance of me, of course, but are those polymorphisms already established, the difference that are prevalent in the population? And why did you choose to work with this HepaRG cell line? Because
Okay.
if one of the impact of this gene in particular is actually the differences that we observe in the population, why choosing this specific cell line?
Okay, so regarding the genetic variants, we start to study and to create artificial mutants, so select specifically mutants that were somehow specialized for specific CYPs. And then we observed that these mutants were in the necessity of other important other important residues that were determinant in the binding. So some of these residues were variants, natural variants found in the population that are established. And this also was very interesting. So probably in different populations or in different subjects, we have different drug metabolism, not only due to CYP polymorphisms, but also due to CPR variants. Secondly, the model, well, we know that HepaRG is a very fine model to study these drug metabolizing enzymes, because the levels of expression and activity during several weeks are fine and easy to maintain. So this was the reason why we select this model.
Yeah, and I'll just say one other reason was, it's advantageous to try and set up a permanent knockout.
Yeah.
We say with a progenitor cell line, because then it's It's much more stable. So that was another reason. You know, HepaRGs are derived from, as you know, everyone probably, from progenitor cell lines. So they form this by progenitor cell lines. So they form the, you know, hepatocyte, cholangiocyte-like lineages.
Yeah. In fact, we can use that progenitor cells to edit specifically the genomes and in the CPR and see the effect of these variants.
Yeah, but one slight problem with the HepaRGs is when you get to the stage like the German group, they did isolation, but it's a pooled isolation, so it wasn't a clonal isolation. So when you try to do a clonal isolation, the problem with HepaRGs, they like each other's company, like in vivo, cells like each other's company. So trying to get a single cell clonal isolation and expansion is quite challenging. That's what we found, you know, with Elisa's work.
Yeah.
But I think there's ways around it. You know, we can transfect with a plasmid reporter and all this sort of things we discussed before, Michelle. And so
I hope I have answered your question, Joanna.
Yes, you did.
Okay.
I mean, I'm just, of course, if we want to study different individuals or the impact of the polymorphism in the population, then we have to maybe to use cells from specific donors. If we want to study the impact of this gene in different, I mean,
but that's more in a population level. We start with the molecular level
that are particularly susceptible or something.
Yeah, of course. Genetic study on a population will be very interesting to perform. It also requires some clinical data, and that's more difficult.
Exactly.
But of course, it's the next level. Yeah.
And the things get more complicated if you think we have two alleles, so that it's going to be interesting.
Yep. But thank you so much, Francisco.
Thank you.
Maribel, do you want to say something?
Sorry, Javier?
No, I was asking Maribel if she wants to add something.
Well, I am really impressed by the work that Michael is doing on cytochrome polymorphisms, variants, particularly understanding the basis of the work, how they performed that work, and try to understand also the impact on drug metabolism and the potential of adverse drug reaction. And I think this is a very important area, because I wanted we are willing to understand what will be other drugs, concomitant drugs that the patient is taking, how they might influence the potential of hepatotoxicity of a culprit compound. No, in that regard, it might modify the concentrations, this plasma concentrations that the patient may achieve more, or because you know, if the drug is during drug metabolism through cytochrome produce a reactive metabolite, in that regard, a variant or a poorly performant cytochrome CPR might preclude the formation of that reactive metabolite. So in that regard, it will operate against the development of TB. Otherwise, if it's the natural compound per se, that the one that is toxic, and if that metabolism is not accomplished properly, then it will be able to have very high concentrations of the drug in blood and produce hepatotoxicity. So I was also very surprised to see that cytochrome at mitochondrial level and also in the reticulum are different. So it has been very interesting talk.
Thank
I'm
you.
really impressed by the work that you are doing, and also the collaboration with Leonard at the cell level, no, with all the potentials that it has.
Thank you, Maribel.
Yes.
Just need some funding now, that's all.
Yeah, that's the main issue.
But we have blood samples from patients with drug-induced liver injury, so I don't know if
Probably we can study that population specifically also. Yeah.
yes, because
Good.
we measure some genetic variants, and we did not find any difference between controls and DILI patients. Particularly, it was in 2C9 and 2C19 drugs that were substrate for those cytochromes. No difference.
Okay. Probably we should study the CPR POR gene.
Yes.
Probably.
I think
Okay, it's an idea.
Yeah.
I think it's also very interesting that what Francisco highlighted is the fact that many other non-cytochrome P450 mediated reactions are dependent on CPR. And I think the heme oxygenase is a very interesting one.
Yes.
Particular of the carbon monoxide, which we know is a gasotransmitter, and also in the iron homeostasis, which is also very interesting because there is a link between ferroptosis and DILI.
Yeah.
So there's a lot of interesting things that we can spin off from this, certainly.
Yeah, for sure.
Yeah, and its potential role in NAFLD as well. You know,
Yeah, that's what I was checking this morning, Leonard, and I found some very interesting things.
Yes.
Keep that quiet just now. Yeah, it's amazing. It's a new field of research. Well, a new field of research.
Yes.
A burgeoning field of research.
Yeah, certainly. Great, Francisco. Good talk.
Thank you. Thank you all for the input also and the questions.
Thanks, Francisco.
Thank you, Francisco.
See you in Edinburgh soon again.
Yeah, hopefully. How's the weather in Edinburgh?
Freezing.
Yeah, okay. Well, here...
Look, I always show, you can see the wind howling and the leaves are blowing off everywhere.
Okay. As you know, here you can see the sun.
And Maribel is sitting in her garden.
Yes. But it's just a background.
Oh,
It's not real.
I thought you were on your balcony or something.
Yes.
Special effects.
Yes.
But today we have 20 degrees centigrade.
Whoa.
So, yes.
And Lisbon, it is also around 19, 20.
Yeah.
Yes.
Nice.
Very
Yeah.
nice.
Edinburgh is about 4.
Well,
I will be leaving just to say goodbye to you.
No,
Bye.
Joanna,
Bye-bye.
I
Thank you, Francisco.
I wanted to tell you,
Thank you,
to
Joanna.
congratulate you also on the training school that you have just finished in October, Joanna.
Ah,
Because
sí.
I have received very nice input from everybody who attended the training school. So congratulations.
Good to
Yes.
know. That's a good feedback because
Yes, it's a lot of work, no? To
Yes.
organize and to make it happen. Yes,
it is.
Joanna.
But if the people like and feel that they learned with that, then it serves the needs. I mean,
Yes.
you feel like you did you work, but for something, for a good reason. It's okay.
I can give my personal testimony again. It was very well organized and it was very well attended. It was very interesting.
Yes,
So
Very nice science. Yeah.
yes.
Indeed.
Triumph.
So that's wonderful.
Yes. And Danielle is back home again.
Yes, and I think he might be somewhere here
Yes.
listening, no, Daniel?
Ah, Danielle.
Yeah,
Yes, I'm here.
yes.
Yeah, I'm back home. Already missing Lisbon a lot.
Yes.
I
But did
want to
not
take,
have much time, because he's been working very hard
yeah,
since he arrived.
And I would like to take this opportunity to thank Prof. Joanna and all her team for having me and for make that experience being such an amazing personal and professional experience. I will always recommend something like this to everybody. So yes.
So Daniel, you should be one of the next ones to present the work.
Of course, yes. I know. I am getting ready to that moment.
Yes. Thank you, Joanna, because yes, he's really amazed. And we will continue, no?
Yes, a lot of work has been done, and I'm sure we will continue to
Still
collaborate
a lot to do.
and to further analyze all the results we have ongoing.
Yes, yes. You know, Fernandez Checa wants to, Carlos, Jose Carlos, if you want to write the question, we can put voice to your question.
Yeah, because he...
Can you read that? Will you be able to compare these outcomes with the PIRF knockout humanized mice? That's the question that is posing Professor Fernandez Checa to you, Francisco, or to any of you, Michael and Leonard.
Well, I'm not sure what is the PRF humanized knockout mouse.
Me neither. I think maybe.
Unfortunately, I cannot talk, but could you? Yes, because his microphone is not working.
These, can you hear me?
Yes.
Yeah, these, I'm just looking at our review again.
Yeah, it's a novel.
These are the mice, humanized mice with the POR, the CPR, the POR human gene knockout.
Okay.
Yeah. And a hepatocyte specific knockout.
And
that was work from, I think, from the lab. This is work that has been done already some years ago.
It's Barzi et al.
No.
Yes, Carl Weissig.
Barzi et al. Yes, I have here Nature Communications.
Well, there's that one, and there's this subsequent one on P450 humanized and human liver chimeric mouse models for studying xenobiotic metabolism.
It will be also interesting to use these mice as a model in the next step after studying and demonstrating the cell lines, hepatic cell lines, that the effect of these variants, if effectively confirm some levels of hepatotoxicity or difference in drug metabolism, maybe we can use these models in future studies to keep in mind.
Yeah, so I'll put the reference up on the chat. So
he has sent by chat the paper.
Yes, you're right.
The paper.
Oh, sorry, it's the Nature Communications paper. I was looking at something else.
Yeah, 2017. Yeah.
Well remembered.
Yes.
Yeah, that's a nice study. So you can look at, reactive metabolites and, difference between SIP-dependent and non-SIP-dependent metabolism if you knock out the POR completely. So it's quite advantageous in preclinical safety assessment.
What surprises me is that the knockout of POR gene did not implicate that the viability of the mice.
Now there is an earlier study. I think it's from the early 2000s. I think it was a work from Dundee. I forgot the name of the author or the main. They made a knockout complete knockout POR, which is not viable. And the only mice, and what they found some litter, some offspring, but they found out it was the POR knockout was leaky, and they made a conditional hepatic knockout, and that was viable, and they could study. So it was from it was from Dundee. I don't remember exactly the name of It's Roland Wolf.
Yes.
He made it's in beginning of the 2000s or something. They did some studies on that.
Yes.
Completing knockout will have impact in the viability of the organism because
of course.
involving
I mean,
steroidogenesis in
exactly. I
vitamin
mean, the
embryonic development.
Yes, the Antley-Bixler syndrome is indicating this very important role of the CPR in development. So,
but in the humanized mice, it's a conditional knockout, so that's why it's not lethal.
Yeah.
So it's liver specific.
Yeah. Probably a model to apply in next projects for funding.
Right. Yes, sure.
We should contact these guys, yeah, for sure.
Yeah.
Why you don't want to reinvent the wheel, you know?
Of course, they already did it.
Yeah. It looks great.
Interesting.
They have experience.
Suggestion it is. Yeah.
Jose Carlos has experience in this model. They did. I don't know if you are still using this model, this humanized murine model, because it was difficult. Was difficult to maintain. Yes, to keep the animals alive. No.
Okay.
Yes, we have.
We have the FRNG mice with humanized hepatocytes and plan to double immunization with hepatocytes. And okay, good. Interesting. Yeah, good, great. That would be an interesting approach.
Yeah, of course, I will contact.
Yes.
Yes. That would be an interesting approach indeed.
Jose, do you have laryngitis?
No, it's the microphone. It's the microphone.
Microphone laryngitis. It's a stream. My Teams has stopped working completely. Just stopped last week on MacBook with Safari. So I don't know if anyone else has had problems. It stopped working. So
yes,
I had to resort to Zoom. It is FUC microphone.
Yes.
I see. That's a terrible brand.
Yeah, not very functional. Okay.
So, Javier. When?
Yes.
It will be the next.
So the next one is like we said, is gonna be the 15th of December. The third, I think is the third Wednesday of it's the 15th of December. And Alejandro Ionides, who is I think is with us now. He will be presenting what he did in Oslo with Gareth Sullivan.
Okay.
Okay, interesting.
Great. Yes.
Great. So we will see you the 15th of December at 4:30 as well.
Perfect.
Okay.
Well,
okay, excellent.
Okay.
Okay. Thank you very much.
Thank you, Javier.
Good to see you, all of you.
Yes.
For the nice discussion.
Bye-bye.
Thank you all.
Bye.
Bye, Lenny.
Thank you.
Bye.
Bye, Lenny. Bye, Michelle.
Francisco.
Bye.
Cheers.
Thank you,
Great
man.
stuff. Really
Bye.
good.
Thank you.
Excellent.
Bye.